Good evening, everyone, and welcome to the first Palm Beach Science Series of the new season. Somebody said to me it's like opening night here, right? It's Thanksgiving week, it's the Monday night, we're all together. For those of you who do not know me, I am Mark Roithmer. I have the privilege and honor of serving as the CEO of the Alzheimer's Drug Discovery Foundation. It is wonderful because we have both faces that we've had before and new faces. Notice I didn't use that three-letter word beginning with an O and D. So it's great to have some new friends and as well as friends as we've seen at the series before. As always, we will start this with a big thank you to Heidi and Tom McWilliams. Yeah. Tom, who serves on our board as the co-vice chair of the overall board and chair of our mission-related investment committee. And Heidi, who is co-chair of all things Palm Beach, but it was really Heidi's vision over 2 years ago. Let's bring the science to Palm Beach, and how right she was. So Heidi, we are indebted to your vision. Thank you. We have a couple of other co-chairs here with us from our board of directors, Ms. Nancy Goods. Nancy, yeah. Who started our Palm Beach Hope on the Horizon event going on 6 years, Nancy. 7. Who's counting, right? Which is great. And also with us tonight is another co-chair, but she's not going to be co-chair of the event this year because we're honoring her this year, is Ms. Bonnie Lautenberg. And that event's taking place March 15th. And I know we all love Bonnie. The other co-chair here for us in Palm Beach is Judy Lauder. And that brings me to the family Lauder. About 25 years ago, Leonard and Ronald Lauder, at the pushing of their mother, Estee, co-founded the Alzheimer's Drug Discovery Foundation based on 3 principles. 1, it would only fund translational science. What does that mean? And so those of you who've been with us before know, how do you go from bench to bedside? How do you go from a good idea in a lab and make sure it's helping and taking care of people? Number 1. Number 2, the organization would not make grants. We would not give out grants. We would make investments. So the money from the foundation that was going to be given out, there would be a contract, whether that was with an academic or whether that was with a biotech. And if that institution saw any return on investment, a portion of that comes back to the ADDF, which we put right back into the science. It's called venture philanthropy. And then the last principle that the Lauder family had was they would partner with anyone to get this done. We have an office of about 28 people, 14 are scientists, 14 are not scientists. And it was to say to other philanthropists, come join us in this journey, in this fight for Alzheimer's. We have the scientists, we want to work together. And I point that out specifically because those of you who've been with us before, you've seen this happen. People like Bill Gates, people like Jeff Bezos, people like Mackenzie Scott have partnered with us now on this journey. The other piece that's important with the Lauder family is the Lauder family takes care of all of the expenses for the Alzheimer's Drug Discovery Foundation, so that anybody who wrote a check to be here tonight, every cent of every nickel of every dime of every dollar goes straight to the scientists and the science on the front lines, right? And that's a debt of gratitude we owe to the Lauder family for that. Which gets me to the science. At the very beginning of the ADDF, and almost 25 years later, we fund 3 things in science. We fund prevention, we fund biomarkers, and we fund therapeutics. And just to give you a sense of that, we now know on this diagnostics and biomarkers, there's a blood test you could take tomorrow that can be predictive of whether or not you're going to get Alzheimer's. We also know that with Alzheimer's, it can predict it 20 to 30 years before symptoms ever take hold. And we also know, those of you who met Dr. Mia Kivipelto last year, you heard her say in these very rooms that 40% of dementia, up to 40% of dementia can be prevented. We can push it out through different lifestyle changes. And the thing that you're going to hear more and more about literally next week and in the coming weeks, there are now therapeutics for the first time in over 20 years that are coming to market that can do something about it. So this whole thing of you can learn that this may be in your life well in advance. You can do things preventively to take care of it, and there's a whole cadre of therapeutics that are literally on the horizon, and you're going to hear about that today. But you're not going to hear about that from me. You're going to hear from two leading experts who we have with us today. I'm going to introduce the first. 25 years ago, when the Lauders started the ADDF, there was a wonderful gerontologist who happened to be a scientist and an MD that they and the family worked with by the name of Dr. Howard Phillott. And Howard is one of the leading experts in the world on Alzheimer's. In fact, he's been doing this so long that at the beginning they called it senility, right, Howard? And now they call it Alzheimer's. The Lauders went to Howard and said, Howard, what do you think of this idea that we start a foundation, translational science, we're going to do investments, not grants? What do you think of all that? He goes, I think that's perfectly spot on. I don't think he was prepared for the second question, which was, Howard, would you leave your day job and come and work for us to lead this foundation, to be the first employee ever? And his day job at the time, he'd been in academia, he had been in industry, he gave that up, but he keeps a small private concierge practice because, as I know, I've worked with Howard now for going on 6 years. He has only one true north. What can all this do for patients and their families, all right? So it is at this point I'm going to turn it over, ladies and gentlemen, to the co-founder and the chief science officer, someone who any of us at the ADF consider a good friend and a deep and respected colleague, Dr. Howard Phillott. Howard?
Thanks, Mark, for a great introduction. I'm not going to dwell on an introduction because Mark has really laid out a great landscape here. I will say one thing. Perhaps coming at this as a geriatrician and a student of aging and an internist rather than a neurologist, I've thought a lot about the fact that we've had research on aging for well over 120 years. And Alzheimer's disease is a disease of aging and old age. And most of the therapeutic approaches to date that you've heard about or that we might discuss a little bit here today are really directed at the neuropathology of the disease, which is the plaques and tangles, but not really until recently directed against the biology or the pathobiology of the disease. And so we sort of took the tack from the very beginning that there's a tremendous amount of knowledge about the biology of aging, but none of it had been translated into developing new therapeutics for old age, for Alzheimer's disease. And a good example of that is inflammation, which is one of the hallmarks of aging. But for many years, there was no real concerted effort at trying to develop therapeutics to reduce the inflammation that even Alzheimer's described in 1906 when he described the first person that had the disease. So we've had a unique point of view over many years. I can say that probably 5 or 6 or 7 years ago, if you looked at most of the drugs in development, they were against amyloid, which is this protein that is present in the plaques, and very little on these other alternative pathways. But today there's over 120 drugs in development, and from our report that we put together recently, over 75% of the drugs that are in development are non-amyloid, non-tangle type of drugs, which gives me great hope because we are on the verge perhaps of seeing the first drug approvals since 2003. So we've had about a 98% failure rate in Alzheimer's disease since about 2003. But for the first time, we're seeing a lot of drugs being developed that are novel. They're directed against innovative mechanisms. We're going to talk about that today. And so that gives us great hope because even if these anti-amyloid drugs come to market, they're only having a modest effect on reducing the rate of decline of the disease, about 30%, let's say, of a change in the rate of decline, which is on the cusp, I would say, of being clinically meaningful, but certainly statistically meaningful and ultimately probably will pass FDA, and we can talk a little bit about that. But what we want to do is not slow the rate of decline of the disease by 30%. We want to slow it by 100%. We want to stop it in its tracks. And I think the way that most of us now have come around to thinking about that is a good analogy is cancer. And if you think about the early days of cancer treatment, doctors were throwing toxins at cancer patients one at a time. And then Sidney Farber and others came up with the idea that maybe we needed combination therapies. And so they started doing combination trials and found out that combinations of drugs worked better than single therapies. And then understanding the biology of the cancer cell, it was recognized that there are many pathways, and by the way, cancer is a disease of aging and old age. They recognized that there were many pathways in the cancer cell that were disrupted, contributing to the tumor formation. And so we developed biomarkers for cancer. And then combination therapies based on the biomarker, something we call today precision medicine. So if you go in and you're coughing, you get a lung tumor on your x-ray. But the biopsy is done, and then the lung tumor itself, the cells are characterized, and then your treatment is tailored to the type of biology that your cells express. And it's been very successful, right? Because now cancer is a manageable disease in many cases. And that's the vision that we have for Alzheimer's disease. We're going to have the first disease-modifying therapies in the history of the world, probably come to market in the next few quarters. And it's going to be very exciting, but it's only the beginning of what I think we consider in the field a new era of therapeutic development for Alzheimer's, which has been enabled by biomarkers the same way in cancer. We're typing these cells, we're looking for biomarkers that characterize patients. Now, we're here today, we're very privileged to have Dr. David Holtzman, who is a professor of neurology at Washington University. We've been colleagues in the field for a long time. And he was chairman of neurology at Washington University Medical School for about 18 years. But I think perhaps one of the most proud things that I would like to say about David is that with an ATDF investment, a modest investment, very early days, David and his group spun out a biotech company called C2N, which was very innovative and risky at the time. And I guess it was probably 10 or 15 years ago that we made that early investment. But I guess in the last year or two, maybe 2 years ago, the technology resulted in the first blood test for Alzheimer's disease. And this is really incredible. If you had said to me 5 years ago, I mean, I've been taking care of people with Alzheimer's for 40 years. We've been doing research on Alzheimer's for 40 years. I've been doing research. If you said to me there was going to be a blood test for Alzheimer's disease 5 years ago, I probably would not really have believed it, and certainly 10 years ago. And I just want to say before I turn it over to David and ask him a question is that what enabled this modern era of having blood tests, in addition to brain scans, amazing brain scans for early diagnosis that we can use, but having a blood test, imagine how that's going to change care for people with Alzheimer's disease. Most people that get a diagnosis of Alzheimer's get it in primary care, but primary care doctors are not good at making a diagnosis of Alzheimer's. They're not doing a lot of cognitive testing, and they're usually referring to neurologists. Imagine if there was a blood test that in the normal process in the workspace of care of a primary care doctor, just like cholesterol, just like hypertension, you do a blood pressure cuff, you can measure hypertension. For all major diseases, diabetes, you can do a blood test. Imagine somebody comes into the office and they have memory problems, and maybe the doctor talks to them for a while. The average primary care visit is about 11 minutes in our country. I spend an hour with patients, I'm sure David does too. Do a little bit of cognitive testing, but you order a blood test, and with about 90% certainty, you can tell that person within a short period of time if they have Alzheimer's disease. And then the workflow from there is what do you do about it? And I'm sure we're going to talk about that. It is not a nihilistic event in the course of diagnosis and treatment. And I'll just stop by saying that I believe that every patient with a memory problem deserves certainty around their diagnosis. It helps with prognosis. And I believe that we all need to start with a certain diagnosis that we're clear about. There was a study that the government sponsored, Medicare, that showed that doctors were wrong in primary care practice and even in dementia care practice about 50% of the time in making the diagnosis of Alzheimer's, and that has consequences for care. So the blood test is a revolution. And so let me turn it over to David, who is one of the inventors. He's a founder of C2N, which brought the first blood test to market. It's on the market now in 49 states. And maybe you could tell us a little bit about the blood test, David.
Thanks. Can you hear me okay? So, yeah, there's been an interesting and long journey. 20 years ago, we actually had some findings in an animal model that was related to Alzheimer's disease, where we were finding that you might be able to see changes in the blood that reflected what was going on in the brain. And then subsequently, a colleague came to my lab and wanted to further investigate this, and one thing led to another, and we ended up developing what then was what's still a pretty fancy way to essentially measure a very, very low abundance protein using a fancy scale called a mass spectrometer. And in doing that, what we're able to do is to see when certain proteins build up in the brain that Howard was talking about that are called plaques and tangles, these proteins get stuck in the brain and they don't come out as well. And so what we're able to do is detect in the blood that they're not coming out of the brain and they're lower than they should be, and that indicates whether or not this amyloid protein is building up. And I think it's going to be very useful because as new treatments are coming out, as Howard was mentioning, there's a treatment you may have seen in the newspapers called lecanemab that the top-line results were announced about 6 weeks ago that while it didn't have a dramatic effect, it seemed to slow cognitive decline by about 30% over a year and a half. And it will probably be the first approved treatment for Alzheimer's disease that affects the course of disease. But you can imagine when treatments like that and other ones are being developed, you're probably not every person, if there's millions of people eligible for this, are going to get a PET scan, which is only available in some places, or spinal fluid testing, which is doable, but for millions of people, it's going to be a lot easier if we can screen people with a blood test and get them the appropriate therapies that they ultimately need.
And imagine trying to develop a drug for heart disease if you didn't have a blood test for cholesterol. Imagine trying to develop a drug for hypertension, right, which affects 65% of people over 65, if you didn't have a blood pressure cuff. You know, that's where we've been with Alzheimer's disease, but it's different now because we have a blood test. Now, it's not only going to affect clinical care, but the average phase 3 trial in Alzheimer's disease costs somewhere around $300 to $400 million. And we helped to bring the first diagnostic test to market, which was a brain scan, PET scan, that can see the amyloid in the brain. That's amazing. Yeah. Imagine, you could I can send a patient down the road to the radiology office, get a brain scan, and by the 24 hours, I can tell them if they have Alzheimer's disease or not. But it costs $8,000. Now, if you're trying to do an anti-amyloid clinical trial, like David was talking about the drug that's coming to market by Biogen Eisai, if $200 million, half of the cost of finding the patients for the clinical trials is in the enrollment and recruitment phase. And the companies have been using the brain scans to find the people that have amyloid in their brain so they can treat them with an anti-amyloid therapy. Now, the blood test is about $1,200, if I'm not mistaken.
Yeah, I'm sure it'll be less than that when insurance pays for it, but yeah, it'll be a lot less than that.
So when we're screening people in the community to find these patients, it's going to be a lot cheaper, a lot less invasive, and a lot less expensive.
And actually, in one of the prevention trials that's now occurring with that same antibody, the company, Eisai, is using the blood test to screen now instead of using amyloid. They use amyloid imaging, but only after they're positive or negative on the blood test. So.
And like many other fields of research, this test has been enabled by modern technology, because the old tests, or some of the tests that are out there being developed by companies like Roche and Fujirebio Bio, which has brought a test to market, they're not as sensitive as this mass spectrometry test, which is much more sensitive and able to do that. So, but all of the tests were enabled by new technology that had the sensitivity to pick up the minute amounts of these molecules in the blood. And I think we're going to have more blood tests developed as we move down the line. Blood tests for inflammation. Part of the blood test that you've developed through your company, C2N, is called PrecivityAD.
Correct, right.
And it's actually a combination of amyloid, which is the predominant protein characteristic of Alzheimer's disease, and another molecule called ApoE. So just moving to therapeutics for a minute and novel approaches to therapeutics that are not amyloid. ApoE, 20% of people, there's three kinds of ApoE, ApoE2, ApoE3, ApoE4. 70% of people have an ApoE3 or ApoE3A, and about 20% of people have an ApoE4. But 60% of people with Alzheimer's disease have an ApoE4. So we've known about this risk factor for almost 25 or 30 years, but we haven't been able to drug it. We know it's kind of a low-hanging fruit. So our foundation, David, has been trying to figure out how we can fix this ApoE4 gene, make it into an ApoE3, or even make it into an ApoE2, which is protective. And it's really interesting, this ApoE molecule and its role in Alzheimer's. And I thought maybe David has a unique program on that, so maybe you could tell us about that.
Sure. So one of the ApoE is interesting in addition to being the strongest genetic risk factor for Alzheimer's disease. It seems to have it play different roles in the course of the disease. So one of the things it does, not surprisingly, like most of the risk factors for the disease, it does affect when somebody develops amyloid deposition in the brain. And that process occurs 20 years or so before people become symptomatic. And I've worked on that, a lot of people have worked on that. But one of the things that's emerged recently is that ApoE also affects the brain's the response of basically it affects inflammation in the brain. So when you start developing these pathologies like amyloid and other changes, there's immune cells in the brain called microglial cells that become they're trying to fight off an infection. They see these proteins building up in the brain and they're trying to fight that off. And in doing so, they secrete different molecules that are damaging to the brain. So what we found recently in the last 7 or 8 years is that if you turn down how much of this ApoE is made in the brain, it suppresses the inflammatory response, and that results in less brain injury. So I think one of the approaches that we've been testing translationally in animals that I think will move into humans in the relatively near future is a way to decrease this ApoE4 gene and protein in the brain. One way to do it is to give what's called, it's a long name, antisense oligonucleotides. Basically, it decreases the level of this protein. There's other ways one could do it as well, but I think it's really worth trying to do this because I think what we don't have now in Alzheimer's treatment is a way to, as Howard said, do anything more than slow the disease a little bit. And I think if we attack these other pathways, especially inflammation, it's very likely we can really add on to what we have now.
Yeah, so two things. There was one of the investigators we're funding at the University of Southampton in the UK, Clive Holmes. He showed, and I think there's other evidence, that people can have this amyloid protein that's characteristic of Alzheimer's in the brain. But if there's no inflammation around that, then those people don't progress very rapidly or at all. And Alzheimer actually described these inflammatory cells that David was talking about in 1906 when he described the first case. So we've known about inflammation for a long time. It's a hallmark of aging. We know that it accelerates the development of clinical symptoms in the disease, and we have no therapeutics for that either. And we've been working on that for a long time. And I have to tell you, we met a lot of resistance in the field because the field was so focused on this anti-amyloid therapy. But today, one of the largest portfolios of targets in the field is inflammation. So it's a really exciting time because I think, again, we're finally moving beyond this sole focus on amyloid, and if we can just get rid of, and we're not being negative on the amyloid, but it's just the first step in this process. And so there are a number of anti-inflammatory drugs being developed. There's a company out there called Elector, which has translated some basic research on another gene that was recently discovered called TREM2. And TREM2 is a receptor on the inflammatory cells that David just talked about, the microglia in the brain. And it's thought that by modulating the receptors, these inflammatory cells do two things. In one very positive way, they're scavengers. They're garbage disposal, waste management cells. They're out there to get rid of the amyloid plaques. And so that's the natural defense against it. But they're also inflammatory cells, and when they get overreacted in the inflammatory pathway, they get damaged. So the challenge for the field is how to manage these cells in treatment to lean them to keep being garbage disposal managers, and lean them away from being inflammatory cells that cause damage. And so TREM2 was identified as one of the second genes of many actually that are being discovered now as risk factors for Alzheimer's disease. And that's being translated, and there's a company called Elector that is actually in phase 2 now that is developing therapeutics for TREM2. I don't know if you want to comment on that.
Well, yeah, also just a personal story about TREM2. So in 2013, there were 2 papers that were published showing that this gene called TREM2 is a strong risk factor for Alzheimer's disease. It's not as common as the ApoE4 allele of ApoE. I think only 1% or so of people have this particular risk mutation. But when this was announced, I looked at this and I go, oh my gosh. So I walked literally downstairs from the 8th floor where I am to the 7th floor to a guy's lab named Marco Colonna, who had cloned the TREM2 gene in 2001. He'd been working on it for a long time, and we immediately started to collaborate. And what Howard said is right. It looks like if you turn it on, this inflammatory effect is kind of good during the early phase of Alzheimer's disease when you're pre-symptomatic, but once you start getting a lot of damage in the brain, you might want to kind of turn it down. So that's exactly the challenge in the field now is when do you tweak it, and how much do you tweak it up, do you tweak it down? So we're trying to do a lot of studies in animals to figure that out before we go hog wild. Although Elector is trying something early, we'll see what happens.
And just as an anecdote, I had a patient several years ago, a beautiful young woman. She was about 45, and she had a horrible progressive dementia with no real history in her family. And we did some genetic testing on her, and it turned out she was ApoE4. She had 2 ApoE4s, so that alone would cause her to have the disease onset about 10 years before most people. So instead of 75, 65, and so much greater risk. But she also had a mutation in the TREM2 gene called R47H. So she had 2 of the main known genetic risk factors for Alzheimer's, and she had a really rapidly progressive dementia that she died at about age 52 from that. And what we're trying to do now, actually, through the foundation and through this particular donor who this patient was his wife, is establish a clinic at the University of Alabama of people with this rare R47H mutation so we can better understand why it's so pathological, why it causes an acceleration of the disease, and sort of from an orphan designation, maybe develop drugs for R47H to fix the R47H gene. But what these 2 stories say something about is you hear a lot about genetics in the newspaper. A new gene was discovered. ApoE4 was discovered like 30 years ago, and it's 30 years later and we still don't have effective therapies. And what I want to say is it's a long way from discovering a gene and developing a therapeutic for that target. It's a very long way, and drug development is really hard.
So actually this having 2 copies of ApoE4 and a TREM2 mutation, it's interesting. Last week we published a paper exactly about that topic, and what happens is when you have that unfortunate combination, these microglial cells, the inflammatory cells, build up a lot of lipids inside of them, and they can't get rid of it. And that's probably somehow leading to this really strong inflammatory response. So that's something that should be ultimately be able to develop a treatment for that, I think.
And I just wanted to add also that this ApoE4, the ApoE protein in general, has many functions. If you do a Wikipedia on it, the list of functions that it seems to have is enormous. So people, including us, were trying to develop drugs based on 1 or 2 or 3 of those pathways that ApoE4 is involved in. And then we sort of realized, and I should say Rod Crystal, who is the chairman of genetics at Weill Cornell and is a very successful bio-entrepreneur, he said, this is a genetic abnormalities. Let's develop gene therapy for it. So what we did was we started funding him at Weill Cornell and helped him spin out a company and invested in the company called Lexeo. And what they're doing is ApoE2 is protective. ApoE4 is the damaged one. If you take people that have 2 ApoE4s, they're the ones that are at greater risk. Imagine if you could change their brain so that their brain was able to make ApoE2, because we know that people who are ApoE2 4, right, they're protected and they have neutral risk. So what we're doing, and what Dr. Crystal is doing at Weill Cornell is he's doing gene therapy for the ApoE4 abnormality. He's taken a virus, it's approved by the FDA, it's a non-infectious virus, and he's putting the ApoE2 gene into this virus. The virus with the ApoE2 gene becomes the drug. Inject the virus into the spinal fluid, it goes throughout the brain, and the brain starts making ApoE2. And he's in phase 1, he's going to report the results at a public meeting next week. He's tested about 5-6 patients at this point. It seems to be a safe therapy. And he can show that with the gene therapy, people who are double E4 make ApoE2 protein. Whether that's going to work to slow the disease down or offset the risk for the ApoE4 risk, we don't know yet. But it's another approach, another novel approach to the whole approach to novel therapeutics. Also, David, there's a strong vascular component to Alzheimer's disease. I thought maybe you want to talk about that because I know you've been interested in that for a long time.
Well, there's two things. I think as people develop cognitive impairment, say over the age of 70, Alzheimer's disease contributes to many people that do develop cognitive impairment, but this probably the second most common cause of damage in the brain, and in sometimes overlapping people is developing small strokes, basically, or just vascular damage. So one thing that's already been shown to, it looks like it's very effective at delaying the onset of dementia is outstanding blood pressure control because it decreases your vascular risk. But in addition to that, one of the other things that happens in many patients with Alzheimer's disease is they develop this amyloid protein we're talking about not only in the brain, but also in the blood vessels. And when that happens, that can also decrease blood flow to the brain. And so just thinking about therapeutics, we don't have good therapies for this problem where amyloid builds up in blood vessels called cerebral amyloid angiopathy. So one of the other things we've also worked on recently is a way to get rid of the cerebral amyloid angiopathy by targeting ApoE, which also contributes to that particular problem. So I think there's a variety of things that could be worked on, some of which are obvious, just prevention, blood pressure control, diet, but also maybe some targeted therapies for depending on what the vascular problem is.
Well, I want to get back to the blood pressure control thing, but also, and the spirit of what you're hearing here is novel approaches to treatment beyond amyloid that you read about in the newspaper. So we funded a study, Professor Strickland at the Rockefeller University several years ago, and he showed that this very potent, very important coagulant protein called fibrinogen, which throughout the body, if you get a cut or whatever, it's very involved in helping you to form a clot. And what he showed was that fibrin, which is a derivative of fibrinogen that's activated by damage to the blood vessels, binds really tightly to the amyloid protein. And these fibrin amyloid complexes deposit in the blood vessels, even the small blood vessels, and he showed this in an animal model. And when they're deposited in the blood vessels, they cause inflammation around the blood vessel. And it's thought that it's possible that one of the ways that you can create a link between systemic vascular disease and the amyloid plaques is the binding of this fibrin to the amyloid, the deposition of these complexes, which are very stable, and the induction of inflammation inside the brain. As a result of that, a company was spun out of UCSF called Thorini, and they're pursuing this strategy of trying to interfere with the fibrin amyloid interaction in blood vessels with a small molecule. We're very invested in this company, and we're very excited about this possible alternative, novel, innovative approach for a number of reasons, not just to reduce the amount of angiopathy in the brain, but also because platelets and these coagulation factors are very involved in the risk of diabetes and hypertension, and even perhaps atherosclerosis as risk factors for Alzheimer's. It kind of creates that link between these systemic diseases and the amyloid plaque formation and the characteristic pathological hallmarks of Alzheimer's disease, which kind of leads me to another novel pathway that's being pursued these days, which you might have heard about, and that's the fact that diabetes is a risk factor for Alzheimer's disease. And so many of you might have heard about metformin, and everybody that I meet wants to be on metformin. And I would say about 10 or 15 years ago, we sponsored a clinical trial of metformin for Alzheimer's disease up at Columbia University, a professor there named Jose Luxinger. And as a result, part of our return on philanthropy is not just investing in biotechs, and we've made, I think, upwards of about 130 investments in small biotechs over the last 25 years. But in our investments in academia, where we've made hundreds of investments in new therapeutics, we're really interested in the metabolic changes, the insulin resistance that occurs with aging, diabetes, and in particular the fact that there's insulin resistance in the brain. And the way I'd like to introduce the topic is to say, what's the first thing that happens when a diabetic gets a low blood sugar because they've taken too much insulin and blood glucose goes below 60? What's the first thing that happens? They go unconscious. The brain is 3% of the body weight, and at any given moment, it's consuming 20 to 25% of the energy that we consume or use through basically glucose and oxygen. So that's kind of an example of how insulin resistance with aging on a chronic basis can lead to neurodegeneration, because these cells are very dependent. They're the most metabolically active cells in the body, the neurons, but they're also at great risk for insulin resistance. And so there's a lot of repurposing going on of diabetes drugs to improve insulin resistance with aging. And this particular doctor just got about a $40 million grant from the government to do a large clinical trial of metformin for Alzheimer's disease. And there's a company called Novo Nordisk, which is based on some work that we funded at the Imperial College of London, using a different kind of diabetes drug called a GLP-1 agonist. And I like to sing this song for you because a lot of you will recognize it if you watch the Daily News with Deborah's permission. If you've ever heard of this drug, Ozempic, it's on the nightly news. It's Ozempic. Okay. Well, we funded some work of a drug related to Ozempic called liraglutide at the Imperial College of London, and Paul Edison there showed that liraglutide would be basically probably good, small trial, for Alzheimer's. And now Novo Nordisk is conducting a 3,000 patient clinical trial of the oral formulation of Ozempic for Alzheimer's disease to reduce this metabolic effect. Now, David, I don't know if you want to comment on diabetes as a risk factor or some of the new therapeutic approaches to that.
Well, I think there's two things with diabetes. One, it obviously also it increases your risk for cardiovascular disease, which affects the brain. But there's also these other effects where it seems to affect brain metabolism, and that's where some of the treatments you're talking about might play a really important role in improving brain metabolism by using some of the diabetic new treatments.
And there was Alzheimer described a novel disease that he named after his friend back in 1906 called Binswanger's disease, which I'm sure very few people, except maybe Bonnie Davis, has heard of. And Binswanger's disease is a dementia, a vascular form of dementia in people with diabetes and hypertension. And we know now that those are the two leading medical risk factors for Alzheimer's. You, I think, alluded earlier to like the Sprint Mind study and prevention and the role of hypertension and as a risk factor for Alzheimer's. Could you elaborate on that?
Yeah, I think, well, this Sprint Mind study that was done looked at people who were late middle age and tried to do relatively tighter blood pressure control. And they clearly showed that the risk of developing very subtle cognitive decline was lower in people over several years if they did the tight control versus not. And what was described years ago, this Binswanger's disease, that's where there was a lot of the area under the cortex called the white matter was really being destroyed by small blood vessel disease from hypertension and diabetes. But what we can see now is that virtually everybody over the age of 60 has some damage to the small vessels. But the question, when it gets a little bit more than just a small amount, that's where we really need to be doing prevention of that initiation phase of small vessel disease. And that's where blood pressure control of diabetes, and maybe other things could probably play a big role in the contribution of that to what we also see with Alzheimer's disease.
So, we have 2 minutes left. Okay, that went fast. Well, then let me just say that we're repurposing hypertension drugs for the treatment of Alzheimer's disease as well, the angiotensin receptor blockers. But we don't have time to talk about that. Yeah, that's what I was going to say. So when we talk about prevention, we're talking about many risk factors. And there's studies that show that probably 40% of the cases of Alzheimer's could be prevented. and there's clinical trials going on of prevention going on. But one of the interesting things that David's been doing is investigating sleep as a risk factor. So maybe you could tell us about that.
Briefly, I think one of the things I'll say is that I was not studying sleep at all until around 2007, when a graduate student in my lab was doing exploratory studies and happened to discover that the different proteins that make up the Alzheimer's disease pathology, like amyloid and tau, they actually fluctuate in the brain with the sleep-wake cycle. They go up the longer you're awake, and they go down when you're sleeping. And that actually turned into a science paper we published, and the implications of this were that when we took animals that developed this pathology and we sleep deprived them, they developed a lot more pathology over a relatively short period of time. We also showed that if you improve their sleep, at this time we used an agent called a orexin receptor antagonist. Now these are on the market to help sleep, that actually suppress the pathology. So I think this is leading to an era where you might be able to test whether promotion of sleep in ways that don't cause a problem might be ways to delay be a prevention for Alzheimer's disease. I think that's what we'd like to test now in humans.
Yeah, we're hoping you'll send us an application so we can fund that trial. But you mentioned these orexin antagonists as a new approach to sleep, and you sort of feel that the drugs like Ambien and that class of drugs is not good for you.
Right, because the classic sleep-promoting drugs like Ambien, they're GABA-A agonists. Basically, they do cause you to sleep, so they're good for that, but you can't, if you take them for a long time, you can really get side effects from them. You can get somewhat addicted to them. Whereas this new class of agents, the first one that came out was Belsomra, but now there's several other ones. Basically, they turn on the sleep center of your brain and not affect all the other cells in the brain. So they just cause you to immediately go to sleep, and they don't have some of the side effects that you see with other agents. So it really might be something worth pursuing.
Are we out of time? Or should we just go to Q&A, Mark?
You get one last question before everybody gets a question. So go ahead.
Well, I mean, there's so many interesting things going on with prevention now, and we think we can prevent maybe 40% of the cases. Some of the new risk factors are like hearing loss, social isolation, and so on. And you were talking about the gut microbiome, and I think that's really an emerging issue now. We're all hearing about the role of the gut and the microbiome in the gut in disease, and there's some evidence it might be involved in Alzheimer's disease.
Right, there's a lot of studies in human health that have shown that in certain disease conditions, the bacteria that are in your gut, which have more bacterial cells than we have human cells in our body in all of us, if you manipulate them, one, they're a little bit different in different diseases, the composition versus somebody who's normal, but that if you manipulate them in different ways, that you can sometimes have health benefits. And our lab has been finding, and we have a paper that will come out very soon on this, that if you alter the gut microbiome in animal models that develop Alzheimer's pathology, that you can strongly suppress the pathology. Now, how to do that in humans in a way that I think it needs to be tested because it's a lot more straightforward. The reason this is probably happening is that the gut microbiome regulates inflammation outside the brain, but then that affects inflammation in the brain, and so that's why this is an exciting new target to think about.
Yeah, I'll just close by saying, Mark, that one thing that David's mentioning is systemic inflammation. And that's a hallmark of aging. Arthritis causes inflammation. All the diseases that we suffer, diabetes, atherosclerosis, these are all diseases of inflammation. And the example I like to give to link the predominance of systemic inflammation with aging and Alzheimer's disease in the brain is, if you think about it, when we teach medical students how to diagnose pneumonia, we look at cough and fever as signs of pneumonia. But when we look at older people that have pneumonia, the first symptom of systemic inflammation from pneumonia is confusion and delirium. Older people don't develop a very effective fever response or a cough, so if you're taking care of an 85-year-old that's developing delirium, one of the first things you want to think about is either pneumonia or a urinary infection, and it's just a really very striking example of how systemic inflammation can trigger the brain and affect brain function.
Good. So we're going to open this up to Q&A, but how about a round of applause? How fabulous is it? David, you're laying down a really good marker for our first in the series this season to how we're going to beat this as we go forward. So just raise your hand and we're going to bring the microphone around and you can ask questions.
First of all, thank you very, very much. I think the whole audience is so appreciative of what you do. So I wanted to continue off of what you were saying about the gut-brain sort of connection. And therapeutically, you were saying therapeutics could be developed to help Alzheimer's. But if you look at it in the other way, because many, I think millions of Americans have Crohn's, IBS, and other related problems with their gut. Is that a risk factor for Alzheimer's?
I don't think we know the answer whether Crohn's is a risk factor for Alzheimer's disease, but one of the things I've found intriguing in the last several years is that when people develop diseases like that now, they actually get treated with agents that suppress the immune response. Things like Humira, other anti-TNF antibodies, things like that. I think that is probably, if anything, would be protective against Alzheimer's disease. But the question of whether the disease itself would put you at risk, I don't think there's good studies that have shown one way or the other that would be the case.
But there was a study of anti-TNF antibodies, of Humira, for Alzheimer's disease, and it was a small study which suggested that if you block TNF-alpha, which is a major inflammatory hormone, call it, from all of these diseases, that you could block the effects of systemic inflammation on the brain. And actually, about 20 years ago, I showed that there's elevated TNF-alpha in the blood of people with Alzheimer's disease. And that's, again, with C2N and new biomarkers, I think one of the challenges that we have today, even through our diagnostics accelerator, which we have a $100 million program with Bill Gates and Jeff Bezos and David Dolby and others like that, to develop new biomarkers. And one of the main foci of that program is looking at these novel inflammatory markers in the blood that promote, as I said before, brain inflammation that's bad for your brain. So regardless whether it's Crohn's disease or whatever, any inflammatory disease or activity in the body affects the brain.
You mentioned briefly tau. How does tau fit into this, and how does tau research fit in?
That's a great question. So in Alzheimer's disease, when you follow people with all these different tests we can do now for research purposes, what looks like happens is that in somebody that ultimately develops dementia from Alzheimer's disease, literally 20 years before that, this protein amyloid starts to accumulate, and it accumulates over 20 years till it gets to its almost as much as ever going to be in the brain. But right before people become symptomatic with memory loss or other cognitive decline, this tau protein starts to build up. And the buildup of the tau protein is what strongly is linked with the damage in the brain. There's a lot of people doing research on tau now, including what is the structure of the protein that changes so that it builds up, what response is it eliciting in the brain that leads to damage. That's one of the main things I'm working on now, is how is it leading to damage, and a lot of the evidence looks like it's stimulating these microglial cells, these inflammatory cells, to damage the brain. But there's a lot of people now, more people now are working on it than previously, that's for sure.
And I would say, if I may, about C2N, what I'm excited about is several companies are trying to develop tau protein diagnostics, which are looking like they might even be better than what's currently on the market through C2N, which is the amyloid blood test. C2N and other companies are going to have tau blood tests, which will be even better.
Yeah, and those are already being shown, C2N's new test as well as other ones. I mean, if you combine the tau marker with the amyloid marker, it's as good as getting spinal fluid analysis for whether you have Alzheimer's changes. It's really, really accurate.
So if I could ask a question, if somebody in the audience or in your practice or whatever wanted to get the C2N test, which is the only test really on the market today, what would they do?
Right now, they have to see a physician that then orders the test. You can't just go and get the test without a physician ordering it. And I think in 49 states, there are places that you can go to a physician and order the test. So, and there's a website and it can tell you the information about where in your area you could potentially obtain the test.
Do all doctors' offices have access to the test, or how is the rollout going?
It's been somewhat slow for clinical purposes because I think the main reason is that for anything that's done in human beings clinically, for most people, they need it to be paid for by their insurance. And a new test like this typically it takes a while to get it approved because you need to go to the FDA, you need to go to Center for Medicare and Medicaid, and it's a very long process to submit all the information to them. But it is available, you can get it done, and so yeah, no, it's you can. If anybody is interested, I can send them information how they can do that.
And I know a common question that I get and you get all the time is, why bother getting the test if you're not symptomatic, let's say? If you're symptomatic, it's a different equation, right? Because you want to have some certainty around the diagnosis, which
that's important, really important.
But what about if your father and mother had Alzheimer's and you're 40, 50 years old? What do you counsel your patients to do?
Right. Well, so two things. One, right now, this blood test or any other test isn't only available clinically if you have symptoms. We're not the company's not even allowed to do it on people who don't have symptoms. Having said that, I think the biggest use of a test like this is actually going to be in midlife when the disease really starts, again, 20 years before the symptoms, because these tests can pick that up right when it starts. And I think in the next few years, it should be a huge impact on people. Let's say you get to a certain age, let's say you hit 40 or 50, you get a test like this. If it's negative, that's great, wait 10 more years, get it again. If it's positive, then you might either start a therapy that might be approved by then, or you get in a trial, so that's where I think it's going to have an even bigger impact.
If one is able to take a test, why wait until there's symptoms? Why not take the test when one is a certain age and do something about it if there is?
Yeah. No, I think why not take the test earlier when you're normal? I completely agree with you, but the regulatory agencies won't allow companies yet to do that. It has nothing to do with, it's just the law. I mean, they won't let.
There's a concept called the value of knowing. It's a health economic concept, and health economists use this to try to quantitate the value of knowing for an individual or a population. So what's the value of knowing to anyone in my practice that I'm counseling where there's, let's say, a family history, and say they're 50, and they say, Doc, I heard there's a test out there, should I take it, right? And so one person may say, I really want to know. So I can do everything I can. I'm going to exercise every day. I'm going to eat a Mediterranean diet. I'm going to sleep like crazy, And the next person, probably more like me, I don't want to know. This is too scary. I'm going to wait 20 years to find out. So I think the rollout of these tests in clinical practice for prevention is going to be within the context of what we call in medicine shared decision-making, where the doctor is there to inform the patient about the value of a test, and the patient is there to express their wishes. And I think so it's not going to be black and white. It's not going to be like cholesterol where the government says everybody should be screened. We're not there yet. But there's a value in knowing.
But it might be there. It might be there in the future. I think that people need to show evidence that it's useful to get the test. And I think once that's done, it will be.
And there are prevention trials going on. Maybe you want to talk about the ADHEAD trial or some of the other trials.
Right. Well, there's two trials that are prevention. One will read out after a 6 to 7 year trial period, which is called the A4 study, which is using one of the anti-A-beta amyloid antibodies from years ago called solanezumab. That will read out in the next, I think, 6 months, actually. I don't know if you know, that came out of my lab, but in any case, what's a really exciting prevention trial, though, is this lecanemab antibody, which will likely be approved in the next few months.
And that's the Biogen antibody.
That's the Eisai Biogen antibody. So that antibody is now being tested in people that are cognitively normal and they get this C2N's blood test. If it's and they have to be between, I think it's 60 and 80 years old, cognitively normal. You get the blood test. If it's positive, then you can get in the trial, and then you get a series of other tests, and then you get randomized to either receive that treatment, this antibody, or not. And it's a 5-year trial. And the goal is to see not just whether the antibody removes amyloid, but whether it delays the onset of any cognitive decline. So.
And, 5 years, if you watch the nightly news, and there's always like these happy people with cancer that are taking some drug, and it's at the very bottom in tiny print. You can hardly read it because it's white on white, and it says, this drug improved life expectancy by 7 months. And then the next screen says, and this drug caused death, vomiting, diarrhea. So the whole time you were on the drug, you were sick. Okay, so here we're talking about possibly delaying the onset of Alzheimer's disease by 5 years. And there's been studies that show that if, really simplistic reasoning here, that if the average age of onset of symptoms from Alzheimer's is 75, and if the average age of death is 80, and we could delay the onset of Alzheimer's disease by just 5 years through what we currently know about exercise and so on, and a drug add-on. So it's like the heart disease prevention. You lead a healthy lifestyle, you manage your diabetes and hypertension, and you take a statin. In this case, maybe it's you do all those things and you take an anti-amyloid antibody. The bottom line is if we could delay, it's a realistic goal, if we could delay the onset of Alzheimer's by 5 years, we would reduce the number of people suffering from Alzheimer's by 50%. And that seems like an achievable goal to those of us in the field, I think.
So.
Any other?
Just a question about funding. How much funding does Alzheimer researchers get from the NIH of the total budget? I know a large percentage goes to, obviously, cancer therapy, et cetera, but
Yeah, so when my mentor, who was sort of the person he won the Pulitzer Prize in 1976 for writing a book called Aging in America, Why Survive, which brought to the attention of the American public and the world, really, what he predicted would be the coming epidemic of Alzheimer's disease. And he went with a woman named Mary Lasker, who was a founder of the National Institute of Health in 1948 through a congressional act. And the two of them went to Congress and said, we need a National Institute of Aging, and we need that institute to focus on Alzheimer's. When David and I went to med school when I went in the early 70s, Alzheimer's were not in my textbook. When I was diagnosing people as an intern and a resident, I never made the diagnosis. I never heard of it till I saw my first patient in a nursing home where I happened to be in 1980. And so when Bob Butler started, he got the Congressional Act, they started the National Institute on Aging, they had a very tiny budget, and he did a survey of how much research was going on in 1976 in our country, at a time when billions of dollars was being spent on the war on cancer and heart disease. And we spent, as a nation, $625,000 on research in Alzheimer's. Most of them were caregiver grants. Now, had to build the field. Okay, when I started in this field in 1980, nothing was known. The amyloid protein wasn't discovered till 1984. Build. So about 5 or 6 or 7 years ago, the National Institute on Aging and the Alzheimer's budget was pretty small. It was like in the order of a few $100 million. And just to give you an idea, today the National Institute on Aging is the third largest institute at the NIH, has a budget of about $3.6 billion. And so we've come a long way, is my story.
Yeah, it's just a little bit less now than cancer, not much. So it's really gone up a lot.
thank you.
Well, how much time do we have?
Ask a few more questions if you like.
Well, I thought if there's no questions, I thought I'd follow up on something practical, actually. We talked a little bit about hypertension as a risk factor, and you talked about white matter hyperintensities. And, when we get the result of an MRI, because we always pretty much do MRIs of the brain on people who we're trying to diagnose, very frequently, the MRI comes back that there's these white matter hyperintensities in the brain. And the radiologists that I work with, they're still saying, this is normal aging or whatever. And we know now that this is not all normal aging, but it's a reflection of what we call microvascular disease of the brain. The blood vessels, you're much better than I am at this, but the blood vessels that supply the brain are the carotid arteries, these huge tubes that come in, and then they dive deep and become smaller and smaller, and they're the end vessels of the blood, and so that's where the vascular pathology is, and maybe it presents slightly differently with subcortical symptoms and so on. But there are ways to diagnose this vascular component and even to use it as a biomarker. And I think the National Institute of Neurological Diseases is doing a good job in trying to promote the effort and research on the microvascular changes that occur with aging, change the practice view out there that these microvascular changes that we see all the time on MRI are benign and nothing, when they're really not. And when we see them along with vascular pathology, people probably progress more rapidly.
Oh, no, I think you're right. I mean, sometimes you see patients where you know that the predominant cognitive change they have is due to this vascular damage, and they present with a different syndrome, and so it's a little, but often it's a real mix, and I think the MRI, in conjunction, of course, with the history and everything, can be very helpful in determining how much that contribution might be to their problem. What the National Neurological Institute is working on a lot is trying to better understand how vascular dysfunction beyond simply these white matter changes might be linked to disease, and whether you can actually target the vasculature in some way therapeutically.
Right. And there are drugs, you mentioned about treating hypertension. One of the things that we're trying to sort out, and I think the field is coming to, is which antihypertensives are best for preventing dementia, and which class. And one of the things that I think is emerging is that the angiotensin receptor blockers, like candesartan and telmisartan, are neuroprotective. And we sponsored a clinical trial in Emory that's going to be reported at the next meeting next week on the value of candesartan in the treatment of people with Alzheimer's disease.
Right. Oh, that's great. I think that those kinds of studies are just now finally coming to fruition. So it's important because so many people are taking these kinds of drugs, and it'd be good to optimize which ones would be best for this purpose. So. And
maybe the last thing over the years, there are companies out there like NeuroQuant and Icometrics, and we started a company called ADMDX about 15 years ago, and they're using artificial intelligence and computerized models to read the MRIs of the brain that you and I are ordering on practically every patient. And so in the old days, and even today, I get a report back from the radiologist, and it says, there's some atrophy in the brain, and it's probably related to normal aging, but we don't really know and can't really compare. But then I always order the NeuroQuant, which is available at my radiologist's office, and they give me quantitative information. And it says, this patient's hippocampus is at 1% of the volume of what should be normal, and this is probably Alzheimer's, just based on the MRI.
Right. No, we do the same thing at our medical center, where because a lot of these methods were also done at Washington University and Barnes Jewish Hospital, they actually, whenever we get an MRI at our medical center, and they know who's getting the MRI, then they will do all of this, all the very, very detailed measurements of all the brain regions quantitatively, just like what you're talking about. It can be very helpful, actually.
And I think that there's this ATN criteria that we're using, amyloid tau, neurodegeneration measured by neurofilament, which I imagine C2N and other companies are going to commercialize along with GFAP, using big words here, and then a V component of the vascular, which we're funding the Mayo Clinic to develop that. Maybe you want to talk about that paradigm.
Well, right. So these biomarkers that we've been discussing, you can now in the blood measure the amyloid component, the tau component. There's another component that indicates how much damage is occurring in the brain, this neurofilament protein. That definitely will be added into these panels for sure. I think many of the other neurodegenerative diseases that are not Alzheimer's disease, so some of the other relatively more frequent diseases are things like what's called Lewy body dementia or Parkinson's-related dementia, or frontotemporal dementias, Pick's disease, and other related disorders. We don't have good biomarkers for those diseases, and I think those need to be developed so that you would get, initially when you come in for a diagnosis, you get a whole panel of things to really nail down, well, I have this disease alone, or these two diseases, and not only do you have them, how far along are you in the disease by how much damage there is ongoing.
Yeah, I'm kind of excited of the day when neurofilament becomes part of it, because it's kind of talking about disease activity. I think we see that in other neurodegenerative. So we can tell people how active their disease is, and it's being used in clinical trials. And maybe GFAP would be a marker of astrocytic inflammation.
Yeah, there's a really dramatic thing happened a few years ago. There's a childhood disease called spinal muscular atrophy where babies are born and within a few days they become almost paralyzed and they die within a year. And the treatment was developed for this disease called nusinerra, and there's now two, but that was the first one. And basically it's a fancy gene therapy that these kids no longer die. They can now live what looks like pretty normally. But one of the ways it was detected that that drug was working is they measured this neurofilament protein, which was very elevated because of the damage in their central nervous system, and then it just plummeted when they got the treatment. We can actually see the same kind of thing in animal models where we block neurodegeneration. This protein, neurofilament in the blood, goes way down. So it correlates very well with how much damage is going on.
Well, I want to thank David for joining us
Oh,
here
hold
from
on,
St. Louis.
Howard.
Oh,
Hold on.
You want to close?
Oh, yeah, we're going to have a couple of questions around this. David, take us out 5 years, 5 to 10 years. Blood tests, Alzheimer's. What do you envision? What do you hope for?
I think probably the most exciting thing that will happen by about 5 to 10 years from now is that the current tests plus additional ones will be available so that you can actually determine many years before that you're actually developing a problem, but you're normal, and then you can do something about it. I think that's where I think things will be, hopefully sooner rather than later. How long it will take, I'm not sure, but we can already detect the disease in the brain with the blood test 20 years before symptoms start. So we ought to be able to take advantage of that. And maybe whether some of these new treatments get approved not just for treating people with symptomatic disease, if they get approved for treating patients pre-symptomatically, then that's going to be a great place to be.
And Howard, kind of same question for you with a different twist. So that's where we're on the blood test 5 to 10 years from now. What are you most excited in the field as a whole? Take us out 5 years.
Well, I think these biomarkers are just going to really expand, and it's going to enable, like we talked about, precision medicine, typing patients according to their biology, and then hopefully developing new therapeutics that are the non-amyloid, non-tau type of drugs. And I do think that one of the major advances that's happened over the last several years is not just the availability of biomarkers to enable the way we do clinical trials, but a tremendous amount of knowledge about how to do clinical trials more precisely, particularly measuring things like cognition. And there is new technologies that we're supporting and are in development through the Diagnostics Accelerator and other things where, you know, in the old days, it took 6 hours of a neuropsychologist to do the neuropsych tests in the clinic or in clinical trials, and it took a lot of work. It was very expensive, and now we can do the same thing on the smartphone. And so what's going to happen is both in the community, there'll be passive capture of cognitive function on a smartphone with artificial intelligence, and that'll be applied to screening people using things like speech and language. So if you remember towards the later days of the Reagan administration, and even sometimes people complaining about President Biden, his speech isn't the same way as it was anymore. And there's technologies now that can pick up very early Alzheimer's disease using speech and language parameters on a smartphone. So we'll be able to pick people up with very early Alzheimer's in the community. It's going to make clinical trials much more effective. And I think, you know, getting back to what I was saying, I think this new era of our efficiency and the preciseness with we're able to do trials at a much less expensive number is going to lead alongside all the advances we're making in recognizing these other pathways. I'm sorry to give you a long answer, Mark, but you know, looking at the fact that 75% of the drugs in development now are non-amyloid, non-tau pathways, the short answer is I think in 5 years we're going to have several drugs to market that are going to address different pathways.
And Howard, just to finish it off, what would that mean for you, patients then versus patients today?
I think we're in a phase of hope. You know, we can offer people hope. In the old days, there was no hope. You know, doctors would say, I'm sorry, you have memory loss, you have Alzheimer's, go home and don't come back. Um, I think we can offer people hope now, that we're having breakthroughs, and I think there'll be treatments. And you've heard a lot about prevention today. I mean, the real goal is prevention, and I think we can offer people tremendous hope about prevention that I never would have imagined 5 or 10 or 15 years ago.
Good. Dr. Henderson, Dr. Phillick, could we have some applause? And Heidi and Tom, I think we're off to a really strong start for this season. Hey, one or two things just at the end here. One, there was a very good question, I think, from this side of the room about NIH funding. And the point I just want to make there, the overwhelming majority or bulk of that NIH funding, it's going to good research, but it's not the precise research you're listening to here. Research that's spinning out academics into biotechs and science that's directly into biotechs, funding these phase 1 and phase 2 clinical trials that are these non-amyloid trials. I bring that up for two reasons, and I wouldn't be doing my job if I didn't. One of them is that the way to most precisely get there at 100 cents on the dollar goes right there is investments and donations and philanthropy to the ADDF. And then those also of you that are here that also invest your dollars in a private sense that you do, these companies, they're moving towards something, but they need to move faster. They need capital. So there's both philanthropic contributions and there's private capital that can go into these countries, and that's not quite precisely where the NIH funding is. So we're thrilled how much more is going into NIH, but the tip of the spear is what you're listening to here. And that tip of the spear, if you take away nothing else from today, was this hope and realization, and it's right in front of us, that these combination therapies, think about this, could push out Alzheimer's and dementia 5 years. And we heard today, average age 81, somebody passing. No offense to anybody over 81 here. But think about that. The humanity saved. You know your children, your grandchildren. Your intellectual capacity stays with you until you pass. And that's just amazing. That's what these two doctors have been bringing to us tonight. That's what the ADDF brings to you. Thank you very much, everybody.